I'm Jennifer Markarian, Manufacturing Editor for Pharmaceutical Technology and BioPharm International. I'm here today with Lisa Caralli, Director of Science and Technology at Catalent. Good morning, Lisa. Thank you for joining me this morning. We're here to talk about the drug development process. So about 6% of molecules make it from discovery to phase 3. What can companies do from a CMC perspective to ensure a molecule's success?
That's a really good question. I actually think the most important thing is to really be choosing the right molecule when you start. There's a lot that we can do from a CMC perspective. We can work on the formulation, we can improve bioavailability, but we have to have the right molecule and understand that that molecule can be a drug. For example, if we do work as formulators and we improve the solubility, which improves the bioavailability, but that drug is not stable, say only at minus 80 degrees, that's a molecule, not necessarily a drug. So we really need to think about how that molecule is going to perform in the vial or in the bottle on the shelf, how it performs when you're preparing to dose to patients, and then how it's going to perform in the body. So we need to think holistically about the program and where it is, not just in development, but all the way through to getting to patients.
What tips can you offer formulators to ensure that the transition to later stage work is a success?
Well, one of the things I always suggest is that we have a really good excipient compatibility. It's kind of the foundation for formulation. And what that can do for us is as we get later and we might need to make some changes to the formulation, we then have kind of a recipe book of what we can do to change the formulation. Because if we were just to randomly change an excipient, we might affect the stability. And what's again important is that we think about, we want to be successful. We want patients to be needing these drugs, and we don't want to do anything to jeopardize getting the drug to patient. We don't want to have stockout situations. So again, think about the big picture, make sure we have a really robust foundation and understanding of our formulation, and that's going to carry us through all the way to commercial and getting it to patients who need the drugs.
Do you have any do's and don'ts for analytical method development?
I love that question because I think we spend a lot of time talking about what methods do, and we really also need to think about what they don't do. So with analytical methods, looking at stability indicating methods, we often use UV as our detection method. And that's great, and it works for a lot of molecules, but it doesn't do everything. Sometimes your molecules can degrade into compounds that don't have UV chromophores. Or what happens if it does degrade, but it has the same UV chromophore as the parent peak. So we really need to understand if we're able to separate and quantitate those species. So what I often suggest to people is that you go ahead and you consider synthesizing key degradants for your product and really understand where do they elute. Can we separate them? Can we quantitate them? And do it earlier in the program if we know that they're critical, so that we know that our methods are working early in development. We can identify them and track them all the way from early phase development all the way through the commercial batches.
I've heard that the development history for a drug could get lost. How does that happen?
I actually think a lot of it has to do with how we communicate now. It's very different than it was 20, 30 years ago. A lot of our information is transferred via email, and that can be an issue because you might be transmitting some very important whys to what you're doing via an email, and that gets lost in your thousands of emails in your inbox. And nobody thinks about taking that information and putting it into a more secure source. So again, communication is one of the issues. And I also think it has to do with how smaller companies maybe are positioning their drug asset. in their minds, they may think that Big Pharma is going to come in, they're going to acquire the asset, and they're just going to redo everything. So why does it matter if we have documentation of what we did? But I think we all need to plan that things don't always go as we think, and we may be the ones actually doing the final development and taking it all the way to commercial. So we really want to have robust documentation of what happened, and more importantly, why it happened.